Microphone is working. It's working.
The space.
I don't think it's the space bar. Okay, I think it's recording. All right, how is the audio on these recordings? Is it okay?
Yeah.
Okay. All right, so last time we started talking about the mitochondria, right? So we were really just getting into how the mitochondria are distributed in the cell, right? And how they kind of rely on fission and fusion to regenerate themselves and modulate where in the cell they need to be, things like that. So now we're going to actually get into the function of the mitochondria, which is the production of ATP. So before you get into that, though, we're going to revisit glycolysis, which does not occur in the mitochondria, occurs solely in the cytoplasm of the cell. And last week, remember, I put up like a kind of an introduction to cell metabolism up there. So we're going to definitely go off of that and develop into that in a little more detail. Glycolysis means literally splitting sugar. Glyco sugar and lysis to split. And it is 10 enzymatic steps, which you do not need to memorize all of them, but you're going to learn some of the key enzymes in them that take a 6-carbon glucose and end up making from that 2 3-carbon molecules of pyruvate, also called pyruvic acid. In order to do that, you have to use a little energy at the beginning of the reaction sequence. So 2 ATP are used. But later, that's called the investment phase of the reactions. Later on in the reaction cycle or sequence, you end up producing 4 ATP, right? And 2 NADH are generated, okay? So you have a net production of 2 ATP per molecule of glucose. That is a very inefficient mechanism to make cellular energy. We need to generate much more ATP than that. So using oxidative phosphorylation, which occurs in the mitochondria, is one way to do that. All right. So how does that NADH get regenerated? Well, the NADH is coming from the mitochondria, so the mitochondria plays a role in that, regenerating that. But if organism lacks mitochondria, they're only relying on this for ATP. They have to have an alternate method of doing that. So fermentation is a way to regenerate the NAD plus to make NADH. All right. So some important things to remember here: the breakdown of glucose into pyruvate, that is a series of oxidation reactions. So that those are always coupled to, right, reduction reactions. So NAD plus converted to NADH is the reduction reaction. All right, but glucose is getting oxidized here. So these are all the reactions that occur. And we don't need to learn them all, but I do want you to be familiar with these enzymes here. So the very first enzyme in the process that modifies glucose is hexokinase. And the reason why this is important is, well, it's one of the first enzymes in the reaction. And all of the, well, not all of them, but hexokinase and phosphofructokinase, PFK1, are regulatory enzymes in this process. So as we're going through the citric acid cycle and glycolysis and all this stuff, there are many feedback mechanisms that regulate this process. If the cell has a bunch of ATP, why is it going to spend energy and use enzymes to make ATP that it doesn't need? If the cell has certain amounts of NADH, right, it's also going to regulate these pathways as well. And the other regulatory molecule is citrate. So citrate we talked about a little bit last time, but involved in the citric acid cycle. So if ATP and citrate levels are high, glycolysis and citric acid cycle slow down. And it's these enzymes, hexokinase and phosphofructokinase, that are sensitive to elevated levels of ATP. That's why it's the rate limiting enzyme there. All right, so both of these, though, are kinases. So you should know that they phosphorylate, and it's glucose that they are phosphorylating, or the intermediate molecule, glucose 6-phosphate. Okay. Glyceraldehyde 3-phosphate. We've talked about this protein before, GAPDH. All right, it's a dehydrogenase. What does that mean? It means it's going to take off hydrogens, right? So it's involved in the production of NADH. Phosphoglycerate kinase and pyruvate kinase, right? These are other phosphorylating enzymes. These are the ones that generate the ATP. Okay, so yeah, they're important. These are important enzymes. So, and there are other ones too. When you take biochem, you'll learn all of these enzymes. So I'm focusing on the priming reactions here, all right, which are adding phosphates, right? See how it adds a phosphate to both sides of the glucose molecule. That's the priming. And then I'm focusing on the energy payoff phase here, where we're producing all this ATP. And also the production of NADH through GAPDH. All of these enzymes are considered housekeeping genes. They're all expressed at very basal, steady rates in all cell types. So when we were talking about Western blotting in lab, remember I had as a loading control on one of the exam questions. What was the loading control for that question? What was the protein? That was the protein we were looking at. What was the loading control protein? It's a cytoskeletal actin, beta-actin. Beta-actin is a very common loading control. So is GAPDH. People will probe for GAPDH in lieu of beta-actin. Beta-tubulin is also a good loading control. All right. So net yield from glycolysis, 2 ATP and 2 NADH and 2 pyruvate per glucose molecule. All right. This is anaerobic. It's not respiration. But fermentation action provides some respiration to the whole series of reactions because it's producing carbon dioxide. Forgetting something here. What is this? Okay, right. So the next section in the next subsection in the section is on the citric acid cycle. But before we do the citric acid cycle, there is actually a step that links glycolysis to the citric acid cycle, but it occurs inside the mitochondria. The whole point of this next step is we want to provide acetyl groups for the citric acid cycle. That's what we're getting. So we want to get to the citric acid cycle. So in order to do that, we're going to take pyruvate and we're going to convert it through a decarboxylation step into acetyl coenzyme A. And that actually produces some carbon dioxide as well. Now, here was like good question. So what do you think that is? Oxidation or reduction? It's pyruvate to acetyl coenzyme A. It is oxidation. It's continuing oxidation. Continuing oxidation. Everything going like the carbon molecules are always being oxidized all the way up to the citric acid cycle. All right, so this enzyme, pyruvate dehydrogenase, this is occurring inside the matrix of the mitochondria, and it's modifying each pyruvate and adding these acetyl groups onto it to make acetyl coenzyme A. Okay. So this process uses a little NAD+, just like glycolysis did. So a little NADH is made. All right, and now we're ready to get into the citric acid cycle. So we're also doubling everything as it's occurring. because remember, glucose made 2 molecules of pyruvic acid, and each pyruvic acid is being decarboxylated into acetyl coenzyme A. Okay, so important enzyme here, pyruvate dehydrogenase complex. All right, the Krebs cycle, or the citric acid cycle, is also called the Krebs cycle, named after the German guy that discovered it. Okay, the citric acid cycle is a cycle, which means that something is going to get regenerated. And what's being regenerated is this carbon molecule, oxoalloacetate. So oxoalloacetate is there at the beginning. It's a 4-carbon molecule, and it's binding to this 2-carbon acetyl coenzyme A. Remember, we're bringing acetyl groups to the citric acid cycle, and this makes a 6-carbon citrate. Okay, so that's really the key event that's occurring in the citric acid cycle, in my opinion. All right, so in terms of the carbon structures that you need to know, I really just want you to know those 3. However, there are a series of enzymes that are regulated throughout the cycle, including citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase as well. But this part of the cycle is the most important. So I want you to know these carbon molecules, oxoalloacetate and citrate. And citrate synthase is adding the acetyl group onto oxoalloacetate. Sorry, it's adding the carbon. So what is the citric acid cycle providing? Why is it there? Well, it's not providing a lot of ATP. It just provides a little bit. It does make a little GTP, and that GTP can be used to make some ATP. What it is providing a lot of, though, is NADH, right? So this thing can go in overdrive within the mitochondrial matrix to provide a lot of NADH, and also this other electron carrier, FADH2. It's these 2 molecules, NADH and FADH2, that are going to be fed into the next stage of what we're talking about, which is the electron transport chain. Okay. So I do want you to be familiar with these enzymes. Isocitrate dehydrogenase. This is the dehydrogenator that is producing NADH. Also, alpha-ketoglutarate dehydrogenase is producing NADH. So this is a heavily regulated enzyme, right? High levels of ATP will inhibit it, slowing down the cycle. High levels of NADH will inhibit it, slowing down the cycle. And also high levels of acetyl coenzyme A will inhibit it. All right. Am I missing anything here? That's the important stuff. All right, so what do we have at this point? Well, we've oxidized the hell out of glucose. We've made 2 acetyl coenzyme A molecules. Those are now getting fed into this cycle that's occurring into the matrix, and it's producing a lot of NADH and FADH2. So we have a buildup of NADH and FADH2 inside the matrix of the mitochondria. That energy that's in those high energy electrons is going to be used in the next section, which is the electron transport chain. But real quick, so what we're talking about here, what we've been talking about is just producing energy from glucose. And glucose is the preferred molecule in the cell. If glucose is around, glucose is going to get oxidized to produce ATP. However, if glucose is not around, the cell has an alternate mechanism to produce ATP. And although it's not preferred, it's much more efficient than glucose metabolism. This is the whole premise of like the Atkins diet, right? You starve your body of carbohydrates, and you will start to break down lipids to produce energy. So the reaction that's doing this is a type of oxidation reaction called beta oxidation. So imagine like a long fatty acid chain, right? Which makes up phospholipids, lipids. So beta oxidation is basically through oxidation, the removal of 2 carbons from that fatty acid chain. Palmitate, for example, has 16 carbons on it. And for every 2 carbons that you remove, you produce 1 acetyl coenzyme A, 1 NADH, and 1 FADH2. All 3 of those things can be used to generate a lot of ATP. So, a palmitate that has 16 carbons, you get 8 cycles out of that through this beta oxidation mechanism. So you can produce far more ATP than oxidizing glucose. But it's not the preferred mechanism, right? Glucose oxidation is preferred. This does occur in the mitochondrial matrix, so all of these enzymes and the lipids have to be imported, and we'll talk about that in the later section. All right, so we have produced a little ATP through glycolysis of glucose, not enough. So now we're going to get into the role of oxygen in the mitochondria, which is aerobic respiration and specifically oxidative phosphorylation. So what this section is going to focus on, one thing that the section is going to focus on are, everything we've been talking about so far with phosphorylation has been a form of phosphorylation called substrate level phosphorylation. We are adding a high energy phosphate onto molecules. And you're doing that through some enzyme. But what we're going to see here, there's a new mechanism of adding phosphates that relies on a proton gradient. And it's called a coupling mechanism that does that. All right, so we made a little ATP, but not enough. And we've made a lot of NADH and FADH2. Okay, so NADH and FADH2 are going to provide the electrons for the electron transport chain. And there's basically 2 stages to this oxidative phosphorylation process, right? So the first one is the chain itself. So the movement of the electrons through the chain, through the different complexes to pump protons from the matrix to the inner membrane space. And then the last step is the activation of ATP synthase through the proton movement from the inner membrane space back into the matrix. Right? Those are the 2 stages that are occurring. And you can see here and how they're spatially adapted within the mitochondria, right? So step 1 is generation of the proton gradient. So here are these complexes that are pushing protons from the matrix into the inner membrane space, creating a proton gradient. And then the second stage is the activation of ATP synthase from that proton gradient. So these hydrogens can't get back into the matrix except through this enzyme. And so it squeezes in there and shoots through there and activates the enzyme. And that's what produces the ATP. Okay. Here's our NADH that's getting produced as a result of the citric acid cycle. Here's our FADH2 that's getting produced as a result of the citric acid cycle. And here's all our ATP that's getting produced from ATP synthase. All right, now you can start appreciating that we've got these 2 different electron carriers here, NADH and FADH2. And you can see this guy's going to this yellow complex, and this one's going to the green one. And that's because they do work through different pathways, and we're going to talk about that. All right. So what occurred in glycolysis is substrate level phosphorylation, right? The way that those ATPs were produced, right? It's the direct transfer of a phosphate group from a high energy substrate and then to ADP. All right, now though we're producing ATP through what's called chemiosmotic coupling. Right, and when this was discovered, it was really big discovery. And you can detect the chemiosmotic gradient in mitochondria. There's a procedure called patch clamp methodology where you actually put a very small clamp on the mitochondria and you can detect the change in pH that's in the inner membrane, and also the electrical activity that's provided there as well. All right, so and that's important to realize at this time. The movement of protons, you're not only changing the pH, you're also changing the electrical gradient as well. So two things are being changed by pumping those protons from the matrix to the inner membrane space. All right. The chemiosmotic coupling uses this. Substrate level phosphorylation, which occurred in glycolysis, is just transferring the phosphate. All right, so let's talk a little bit more about chemiosmotic coupling. All right, so two things are being generated. You're generating a pH gradient and an electrical gradient. All right, the protons are moving from the matrix to the inner membrane space through those different proton pumping complexes, and then they can flow back through the inner membrane only through ATP synthase. All right, all of the ATP that's produced in the mitochondria is produced from this enzyme. So you can probably assume that there's a lot of that enzyme found in the inner membrane. And that's why the inner membrane is all folded up to increase surface area, to provide more of these complexes and ATP synthase to maximize the production of ATP. All right, so here are your complexes. Complex 1 is a dehydrogenase. It's going to receive an electron from NADH. Complex 2 is also a dehydrogenase. It's going to complete, I can't talk. It's going to receive an electron, not from NADH, but FADH2. All right, so like I was saying, there are two pathways to do the electron transport chain. There's one pathway starts with NADH, the other pathway starts with FADH2, but they're both going to converge at the same point. So it makes learning this not as bad as it's going to look. All right, NADH gets fed into complex 1, FADH2 gets fed into complex 2, and then they get carried by coenzyme Q10 and they go into complex 3 and complex 4. Okay. So there is a complex 5, just note for like notation, that is ATP synthase. All right, and it's part of oxidative phosphorylation, but it's not directly part of the electron transport chain. When we're talking about the electron transport chain, we're talking about the movement of electrons from one complex to another, and it just includes complexes 1 through 4. Okay, and I'll get into these electron carriers in a little bit. These things, coenzyme Q and cytochrome C. Okay, so here are the two entry pathways, like I was saying. Right, the oxidation of NADH, the oxidation of FADH2. Right, we already did our reduction reaction in the Krebs cycle. Right, as we were oxidizing oxoalloacetate and citrate into all those things. Right, I'm sorry, as we were reducing those things, we were oxidizing NADH and FADH2. Okay, so NADH is going to feed into complex 1, FADH is going to feed into complex 2. Now they both converge in coenzyme Q10, right? So they come out of complex 1 or they come out of complex 2, and coenzyme Q picks them up and takes them into complex 3. All right, so here I've got the first two steps in this list and also this list, right, depend on the complex that's starting the chain. In this one, it's complex 1, which is NADH dehydrogenase. It's only going to work on oxidized NADH. All right, let's just look at the thing here. All right, so here's complex 1. Where is this hydrogen coming from? What area is this? The matrix, right? All right, so where's the electron coming from if this is complex 1? NADH, right? Where's the NADH coming from? Good. Okay, so 2 electrons get into complex 1, and complex 1 is a proton pump. It's going to push hydrogens, right? This is something to remember. In the FADH2 pathway, where it comes in is complex 2, and that's not a proton pump. That's going to be important. We'll go over that in a little bit. All right, the electrons then get transferred to coenzyme Q, and they get made into this QH intermediate. Okay. Get transferred to complex 3. Complex 3 is a proton pump. We've moved 4 protons in complex 1 and 4 protons in complex 3. The electron gets transferred into cytochrome C. The cytochrome C is an important electron carrier, not only in the electron transport chain, it's also important for cell signaling. So when the mitochondria, if they're having trouble and they're under stress, cytochrome C and their other proteins, SMAC and Diablo, will all get released from the mitochondria and initiate apoptotic pathways, so the cell kills itself. So it's not only important in electron carrying, but also in cell signaling as well. All right, cytochrome C brings the electrons to complex 4. So where's ATP synthase in this figure? It's a trick question. It's not in this figure, right? This is the electron transport chain for NADH reduction, right? Complex 5, which is ATP synthase, is not in the figure. All we're describing here is the production of the proton gradient, right? Complex 4 is a little less efficient. It can only pump 2 protons, but these electrons leave, and it also catalyzes the production of water from oxygen. So, this is the sole role of oxygen in the body. That's it. This is all it does. Everything else it does is bad. It's oxidizing, so it degrades DNA, it causes mutations, all that stuff. But if you don't have this here, this whole thing backs up, and you don't produce any gradient, and you don't produce enough ATP to survive. That's why there are inhibitors of complex 4, such as I just drew a blank. It's a famous chemical, strychnine or something. I don't know. It kills you really quick, and it does that through cyanide. Thank you. Cyanide is an inhibitor of complex 4, and it kills you pretty quick because you need this. Cells, remember, ATP is cellular currency, right? Cells use it up quick. They make it quick. You inhibit this, you're gone. Okay. Thank you. Good. +5 there for remembering that. All right. This is the NADH pathway. The other one is the FADH2 pathway. Where did FADH2 come from? The citric acid cycle, right? Very good. So this associates with complex 2, okay? Complex 2 does not pump protons, so look at that. So all the proton pumping we get in the metabolism of FADH2 comes from complex 3 and complex 4. So ultimately, where do you think we're going to get more ATP from NADH or FADH2? From NADH. And that is true because NADH produces more of a proton gradient than FADH2. All right, but everything else is the same here. The electron comes out with the electron carrier coenzyme Q, gets transferred to QH, an intermediate, then brought to complex 3. You pump some protons, electron interacts with cytochrome C, and is brought to complex 4, and you make water from oxygen. Okay. All right, so that's the FADH2. All right, so now we've generated our proton gradient. The proton gradient is producing 2 things: a chemical potential, right, through the presence of those protons, right? That means the environment of the intermembrane space is slightly more acidic than the matrix. And we've produced an electrical potential, right? We have an accumulation of positive charges in that intermembrane space as compared to the matrix. So this produces what's known as a proton motive force, right? These things want to go from an area of high concentration to low concentration. They want to go back into the matrix, but they can't. The only way they can do that is through ATP synthase. So what's we went over this last time. What's the, I'll wait to go over that. Okay. All right, so here's our intermembrane space, right? Here's the cytoplasm. Here's the outer membrane. Here's the inner membrane. And we've accumulated hydrogens in the inner membrane space, right? That's creating a pH gradient and also creating an electrical potential, right? Accumulation of positive charges here and negative charges here. Those protons cannot get back through the membrane, but they can go through what's called complex 5, which is ATP synthase. This is the enzyme that could catalyze phosphorylation of ADP to ATP. And this is not an example of substrate level phosphorylation, which we've been talking about that occurs in glycolysis, right? This is different because the energy that's being used to move that phosphate is being acquired from this proton motive force. Right? So in order to understand how that works, it helps to see what ATP synthase looks like. It has this barrel structure that's embedded in the inner membrane that's rotational. Right? So the FO subunit. And it's you can see the hydrogens can come through here and they cause it to spin, right? The central stalk ends up rotating. And this is like a good example of like a nanobiological machinery. The rotational energy is captured and you can phosphorylate ADP from that. All right. So this is the catalytic region. And this is the embedded rotational region. All right, so all that hydrogen movement is enough to provide a lot of rotational energy to produce on the order of about 32 to 34, just depends what species you're looking at, 32 to 34 ATP molecules per glucose. All right. So what have we talked about here? Well, let me do it here. So we've started out with pyruvate, which is a product of glycolysis, right? Or fatty acids, right, can come into the mitochondrial matrix. You can get acetyl coenzyme made from both pyruvic acid and beta oxidation of lipids. All right, the acetyl coenzyme A gets run through the citric acid cycle. Right, there are decarboxylations that occur, but you ultimately generate a lot of NADH and FADH2. All right, that NADH and FADH2 provides electrons in two different pathways through the complexes that are in the membrane. Some of those complexes pump protons, some do not. And there are also electron carriers that move the electrons from one complex to another. NADH is involved in complex 1, FADH2 is involved in complex 2, and they both feed electrons into complex 3 and 4. Complex 5 is or is not part of the electron transport chain. It is not. It's ATP synthase, does its own thing. All right. Citric acid cycle, producing NADH and FADH2, a little carbon dioxide. This is where most, not all the carbon dioxide that we respire out gets produced. Where does some of the other carbon dioxide get produced? Fermentation. So these things, FADH2 and NADH, are providing electrons to the complexes. They move from one complex to another, pumping protons. These protons accumulate in the inner membrane, and they leak back through ATP synthase, catalyzing the production of ATP from ADP. All right, how you feeling about that? All right, now you guys are experts in cellular metabolism. So why don't you take a 5-minute break, and then we'll do this last section, which is not too long, and we'll be out of here early today, kinda. I can find a cool picture to show you guys if we can find it. Is that the paper? And I'm surprised it's not on the internet. Oh, try spelling it right, that might help. Let's see it. That's not the mitochondria. The hell is that? ER or something. Well, there's a pretty cool image that came out last year in the Nature publication of the inner mitochondrial membrane as seen through transmission cryo-electron microscopy. And it shows like just all of the subunits, all of the complexes and ATP synthase and just how packed in they are in the inner membrane. It's pretty amazing. I'll try to download the paper when I have on my computer, I have access to it, and I'll post it for you guys on. It's not your test, it's just a pretty cool picture. Okay, ready to get this done? Okay. All right, the last section that we're gonna go into today, but not in the chapter. So section 12.5 is chloroplast. And so photosynthesis is pretty cool, but I'm just gonna skip over it. I think you guys have enough to learn with mitochondria. So we're not going to get into the chloroplast, but there's a lot of similarities, like between photosynthesis and the electron transport chain, right? There's many similarities, and so we're not going to do it. But 12.6, we are going to do peroxisomes, and like I said, I may go ahead and record at home a little lecture just to keep us caught up. So be looking for that. And then when we get into we'll just start off with chapter 13 next week. Okay. All right. This section, assembly and maintenance, really has to do with import. So the mitochondria does have its own genome. but it doesn't provide much. Most of those bacterial genes have been taken out over evolution. In fact, I think it's only like 13 or 14 genes that get transcribed and translated by the mitochondrial genome. 99% of all of the enzymes and proteins that the mitochondria require have to be imported from free floating ribosomes that are making these proteins. Yeah, 13 proteins. So those 13 proteins that are encoded in the mitochondrial genome are strictly used for those complexes 1, 3, 4, and 5. It also contains genes for ribosomes because there is translation is occurring in the matrix of the mitochondria. And also tRNAs that are needed to do translation. The genetic code is slightly altered in mitochondria. You can see like UGA, which is a universal stop codon for nuclear genes. It is actually the mitochondrial, the mitochondrial codon for tryptophan. So you don't need to memorize these changes, just know that there are differences in the code. There's this D-loop region on the circular chromosome. That's not chromosome. On the circular DNA that's in the mitochondria. It contains the origin of replication. And that's pretty much it. That's all you need to know about mitochondrial genome. Mitochondrial genomes are important in not parenting, but doing parental screening or whatever, trying to determine what was that show? You're Not the Father.
Oh, Jerry.
It wasn't Jerry Springer. It's like Jerry Springer. Montel Williams. Anyway, Instagram's got a whole thing now going. You're not the father with dogs and cats. It's pretty funny. You guys got if you haven't seen that, it's out there. Anyways, if you're doing like parental typing, mitochondrial genome is a great way to do that. Because mitochondria in us only come from who? Mommy, right? Okay, so we have to get nuclear encoded proteins to the mitochondria. And just like there were different mechanisms to get proteins into the endoplasmic reticulum, there are some different mechanisms to get proteins into the mitochondria. Why do we have to get them there? Well, to do the citric acid cycle, to replicate the mitochondrial genome, to do transcription and translation, right? So this is evolutionary biologists love to study the mitochondrial genome because so many genes have been taken out. Why have they phased out of evolution? So just like in the nucleus, how we had the nuclear pore complex, and in the endoplasmic reticulum, we had the translocon, we have major proteins that are involved in protein import in the mitochondria. Because we have two membranes, we're going to have two different families of proteins. We've got the TOM proteins, which are abbreviated for translocase of the outer membrane. Those are found in the outer membrane of the mitochondria. And then we have the TIM proteins, which are translocase of the inner membrane. All right. So these are embedded in the inner membrane. These are like big beta barrel proteins that embed, and some of them have lateral gates, like we talked about with the translocon, able to move proteins with certain signal sequences directly into the lipid bilayer. And then in some instances, you have these sorting complexes, which are abbreviated SAM, sorting and assembly machinery complexes. All right, so I think we've got 3 or 4 different scenarios that you're going to learn. And I got a little table at the end to kind of sum it all up. So the first one is bringing proteins to the matrix or the inner membrane that have mitochondrial localization pre-sequences. Okay, so these are proteins that have a bunch of positively charged amino acids at the end of the protein. right? They're basically going to first bring it to the Tom complex, and then they bring it to the Tim complex. Right, so look up here. We've got a protein produced in a free ribosome. Why isn't it produced in a ribosome that's bound to the ER? What's happening with those proteins that are produced by ribosomes associated with the ER? Very good. Yeah, remember, those are proteins that are being sent to that are going to stay in the ER or that are going to the trans-Golgi complex to get shipped or to be secreted, right? So all proteins that are coming to the mitochondria are coming from free-floating ribosomes. Very good, Luna. Was the ER lecture, did I post that online only or something? Have you guys seen that yet? Be sure you get on that. It's a big chapter. I think we did that one in person. All right, anyways, so proteins are made on free floating ribosomes that are designated for the mitochondria. So they will interact with heat shock protein 70 to kind of keep it unfolded so that it can pass through the Tom complex. And we're talking about sequences that have this high area of positive charge that can actually associate with some of the subunits in the Tom complex. All right, the protein comes in. In this instance, there's no heat shock proteins that are taking the protein in the intermembrane space, right? So they're really just going right from Tom to Tim. But once they pass through Tim, you can see there are heat shock proteins that'll take the protein and keep it unfolded so that either a chaperonin or other heat shock proteins can help correctly fold it. Okay. All right, so what else is happening here? That pre-sequence does get cleaved. There's a matrix processing peptidase that's found down here that will take off that guy right there. That does require some ATP. And that's really it. It's not much more to it than that. ATP is used to bring the things in. You got to use some ATP. There's two scenarios here. So the protein can either pass through a lateral gate of the Tim complex. This is if it has one transmembrane region. That's an important distinction. You'll see how that is different later on. If it has multiple transmembrane domains, it's passed through a different mechanism. Or it can come out to remain in the matrix as part of whatever's going on in there. Translation or the citric acid cycle. Okay, so that involves a pre-sequence, that positively charged area that's found on the end of the protein. There is also pre-sequence independent import, right? But we're going to the same place. We're still going to the matrix or the inner membrane. Without the pre-sequence, though, there are these other chaperone proteins, TIM9 and TIM10. So these proteins are found in the intermembrane space that take the protein that's been brought in through Tom and deliver it to Tim. All right. I'm not going to get too crazy with you guys remembering TIM23 and TIM22, okay? But, notice that these are different pathways that are going on here, but those can be difficult to remember. What do you think the 22 and 23 have to do with? Very good. Yeah, so the number of kilodaltons that the complex is. It's not like the number of different TIMs that are out there. That would be crazy, right? I think that would be even too complex for evolution to produce, but it has to do with the size of the complex. All right, so this protein does not have that pre-sequence. So there is a signal that's internal in the protein that is directing it to Tom. And in this case, you can have a protein that has multiple transmembrane domains. This TIM complex has a lateral gate. It can pass things through so that you can get the correct folding. I don't know, what could this protein be right here? Just give me something that we've learned so far. What could this be? What do you think? What is this? It's the inner membrane. What's going on here? Electron transport chain, right? ATP synthase. This could be part of ATP synthase. We know complex 1, 2, 3, and 4 is produced directly from the mitochondrial genome. But this could be a number of things, but ATP synthase is one. There are other proteins in mitochondrial membranes. There are voltage-gated proteins and whatnot. So this isn't just, the processes that we've learned so far are not the only things that are occurring in the mitochondria. All right, there's also a way for proteins that are made in the mitochondria, right, through its own genome and its own translation machinery to get embedded in the mitochondrial membrane. And this is something very well studied by evolutionary biologists as well, because this OXA1 translocase is a bacterial protein. It's a bacterial transmembrane importing protein. So, so proteins made in the matrix, you can see, can associate with this translocase and get embedded. Now, this could be any of the complexes that are getting produced because it's coming from the matrix. All right, the last set is regarding proteins that are getting to the intermembrane space and the outer membrane. All right, so here we've got proteins that are coming in from nuclear free-floating ribosomes, right? They're interacting with HSP70, and there is a complex called MIM1 that can take it and push it into the membrane. Or the Tom complex, like we talked about, can associate with chaperones inside of the inner membrane space. Okay, so here's the SAM complex that I was talking about. Remember, we've talked about Tim and Tom. Also now this MIM1, I didn't include it in the major list of proteins because it's really not. But this sorting complex, abbreviated SAM, can take proteins that import into through Tom and can fold them up into the beta barrel proteins in the outer membrane. Good example of this would be like a voltage-gated protein, some big protein like that, that pumps calcium and things like that into and out of the mitochondria. All right, so you've got a few different scenarios here. Ones that are going to the matrix, proteins that are going to the inner membrane, or proteins that are going to the outer membrane. All right, SAM is only involved in proteins that are going to the outer membrane. See that? The TIM proteins are involved in proteins that are going to the matrix and the inner membrane. And the presence of a pre-sequence, you can see here, delivers proteins to the matrix and the inner membrane, right? So this is a lot here, you know that. Be sure that you guys understand what's going to where. If I were to describe the protein, if I were to describe the importing proteins and whatnot. Okay. Are we good? All right. Okay. How do mitochondria get phospholipids? Where do they come from? So mitochondrial lipids are imported rather than synthesized internally. Where are lipids made in the cell? Smooth ER, right? So there's actually a mechanism that can pull lipids from the smooth ER and deliver it directly into the mitochondria. There's also that key lipid component that we talked about, cardiolipin, right? Cardiolipin has a unique, like, double phospholipid structure. It makes up the inner membrane, helps give it extra strength and also extra resistance to protons going across it. It helps support all of those respiratory complexes, and it also helps the folding of the create the folding of the inner membrane to maximize surface area. All right, but these specific molecules that we're talking about here are the phospholipid transfer proteins. which can actually grab phospholipids from the ER and scooch them on over to the mitochondria to build the outer membrane, the outer leaflet of the outer membrane. So that's going to mean that the outer leaflet is growing more quickly than the inner leaflet. How can the cell overcome that? How can it move phospholipids from here to here? We talked about it in the ER. Lipases, right? There are enzymes that can move these from here to here. All right, the last thing we're going to talk about, and we're done. How are other things moved into and out of the mitochondria? Right, so the movement of other molecules actually is mediated through the electrochemical gradient. For example, how does ATP move? Well, we're producing massive amounts of ATP in the matrix. We've got to get that stuff out. So ATP is moved by exploiting the voltage component that's provided as a result of proton pumping. You have an increase of positive charges in the inner membrane space, an increase of negative charges in the matrix. That charge differential will move ATP into the inner membrane space, right? And ATP has a greater negative charge than ADP does. So it's going to move that way. The pH component also helps move substances, right? So we have a lower pH in the inner membrane space than we do in the matrix. And it can assist in moving pyruvate and hydroxide ions as well. Okay? Also phosphate. So that change in pH is responsible for moving those things. All right, you guys, lots to study. Study hard. Remember, Monday, we are not meeting for lab. We are meeting, but we're meeting online, live at 10 a.m. It's a really good idea. Maybe just kind of get going on the lab report a little bit. Just read through that lab report guide. That way you're coming with, some clue as to what we're doing, and maybe you can have some questions and whatnot. Yes, sir.
So for our genetics lab next week, is that still meeting?
We're still meeting.
Okay, so
See you there for genetics.